Welcome to a critical update in oncology research. We are diving into how lipid metabolism significantly impacts immunotherapy response in GI cancers. Let's first understand the design and objective of this groundbreaking clinical study. The study focused on patients with refractory gastrointestinal cancers, meaning advanced disease, where treatment options were severely limited. The treatment involved a combination of camrelizumab, an anti-PD-1 antibody, plus metronomic capecitabine, a continuous low-dose chemotherapy. Metronomic dosing was chosen to minimize immunosuppression and synergize with the immune checkpoint inhibitor. The study's exploratory purpose was to carefully measure host lipid metabolism and body composition. Investigators assessed plasma lipid profiles, including total cholesterol, LDL-C, HDL-C, and triglycerides, providing a snapshot of systemic metabolic health. CT scans were used to measure body composition metrics, such as visceral adipose tissue, skeletal muscle mass, and indices for sarcopenia and adiposity. What did the clinical data reveal about the correlation between metabolic status and treatment outcomes? Patients with more favorable lipid profiles and preserved muscle and fat mass showed higher objective response rates, longer progression-free survival, and longer overall survival. Conversely, patients considered metabolically exhausted, characterized by very low cholesterol, severe cachexia, and sarcopenia, responded worse to PD-1 blockade. This association is not arbitrary. There's a clear biological explanation for why lipids matter for immune function. Activated CD8+ T cells, the powerhouse of anti-cancer immunity, are highly dependent on lipids for crucial functions like cell membrane formation and immune synapse stability. When systemic lipid availability is too low, T cells fail to proliferate effectively, increasing exhaustion and rendering PD-1 blockade less effective. Advanced GI cancers often induce severe metabolic wasting, known as cachexia, which chronically suppresses adaptive immunity and limits the benefit of checkpoint inhibitors. Why does this clinical finding matter for future cancer treatment? This study helps explain why two patients with seemingly identical tumors might have drastically different responses to immunotherapy. One may have preserved metabolic reserves, while the other is metabolically depleted. It provides a strong rationale for adjunct therapies that aim to normalize lipid metabolism or improve immune cell lipid handling, such as exploring the potential of PCSK9 inhibitors. To summarize the core takeaway from this extensive clinical research, immunotherapy efficacy depends not just on the tumor's biology, but critically on the patient's systemic lipid metabolic state. This provides human clinical evidence that lipid metabolism is a legitimate therapeutic axis in oncology, bridging cardiometabolic and cancer therapeutics.